During resting potential, there are more positive ions on the outside and more negative ions on the inside of the neuron. There are more sodium ions outside of the neuron and more potassium ions on the inside of the neuron during resting potential. If a stimulus causes the cell to reach its threshold, this can lead to action potential. An action potential can originate at the axon hillock. The threshold is either reached, causing an action potential to occur, or not reached, so it doesn't occur. This is known as an all or none response. When the action potential reaches the axon terminal, it triggers the channels to open. Sodium channels begin to open along the axon and sodium rushes in. Potassium channels open as well, and potassium is repelled out by the sodium. This is the process of depolarization, which results in excitation. Hyperpolarization can also occur as well, making the resting potential more negative, resulting in inhibition. Ionotropic receptors can also let sodium and potassium ions pass through. This can cause a graded potential. The process of repolarization then occurs. Sodium channels close and sodium leaves. Potassium channels close and potassium stops leaving. The sodium-potassium pump pumps out excessive amounts of sodium and brings in the potassium that was lost. The absolute refractory period then occurs where another action potential cannot be produced. The relative refractory period then occurs where the neuron membrane is hyperpolarized. During this time, it is difficult to have an action potential, but not impossible. There are two neurons involved in neural transmission. A signal is sent from the presynaptic neuron to the postsynaptic neuron. Neurotransmitters are located in the vesicles in the presynaptic neuron. An action potential causes the vesicles to fuse, releasing the contents into the synapse. Another region is called the gap junction, where neurons touch other neurons. When neurotransmitters are released from the presynaptic neuron, it is known as exocytosis. The neurotransmitters then bind with the receptors. The neurotransmitters act as ligands. The neurotransmitters may also be pulled back into the presynaptic neuron through transporters in a process called reuptake. When an action potential occurs, the neurotransmitters leave the receptors to allow the signals to travel. When several synapses receive signals across the entire soma, with dendrites also receiving signals, this is known as spatial summation. When the same receptor and dendrite receive input several times in a short amount of time, this is known as temporal summation. When a neurotransmitter increases the likelihood that a postsynaptic neuron will send a signal, this is known as excitatory postsynaptic potential. Whereas if a neurotransmitter decreases the likelihood that a postsynaptic neuron will send a signal, this is known as inhibitory postsynaptic potential.